Hello everyone, my name is Daniel and I'm joined by Yu and Jiayi. Today we'll be speaking about radiotherapy's effects on cancer-associated fibroblasts of colorectal cancer and how Western blotting was used to study it. We'll be going through an introduction, technique, key findings, impact and significance, and limitations.
Okay, now I will introduce the article first. Going through this article, I was able to conclude that it centers on radiotherapy activated cancer-associated fibroblast growth in enhancing the progressing of tumor through the activation of paracrine in colorectal cancer. In that context, this article allows for the understanding of the way radiation impacts the microenvironment of tumor, resulting in the alteration in the growth and survival of cancer cells. The outcome of this study, therefore, contributes to a broader field by stressing the significance of comprehending tumor-stromal interactions, as well as the importance of establishing other therapeutic intervention techniques to deter the progression of cancer.
How does Western blot work and what are its limitations? Within a Western blot, concentration of a protein of interest is measured within healthy and affected cells, alongside purified solutions of the protein of interest. Proteins within the cells are separated using gel electrophoresis and then transferred to a protein binding membrane, which is treated with primary and secondary antibodies to indicate the presence and levels of the protein of interest in the cells. Blocking solutions are used to prevent non-specific binding, such that the antibodies only bind to the protein of interest. The primary antibody binds to the protein of interest, and then the secondary antibody binds to the primary, giving visual indication on the membrane. Tomalin et al. Used Western blot to detect the activation of insulin-like growth factor 1 receptor and
That's fine.
Tomalin et al. Used Western blot to detect the activation of insulin growth factor 1. Oh, I messed it up again. Oh my goodness. Tomalin et al. Used Western blot to detect the activation of insulin-like growth factor 1 receptor and the downstream mediator ACT. Expression of IGF-1R has been previously related to radioresistance and thus chosen as the predictive factor for radiotherapy outcomes. IGF-1R activates the PI3K cascade that leads to P70S6K activation and protein synthesis. ACT interacts with the B-cell leukemia protein, BCL-associated death promoter complex, inactivating it and ultimately preventing apoptosis, leading to increased survival for the cancer. Colo 320 DM cells were sourced from rectal adenocarcinoma tumor samples, which were then later irradiated and frozen to detect radiotherapy's effect on the cancer-associated fibroblasts. The healthy cells to compare the tumor cells against were sourced from the healthy tissue of 60 rectal cancer patients. A series of primary antibodies was used for detection of IGF-1R, whereas only one primary antibody was used for the detection of ACT. Following a protocol from De Wever et al., the blotting was performed 3 times.
So based on the experiment method that Daniel said, lead to my next point, the key findings about how to use Western blot to get the data and what the conclusions of this data are. And so the first key findings is the expression of the CAF marker alpha SMA is not influenced by radiation exposure. So in the fractionation experiment, the experimenter exposed CAF to different concentrations of radiation. When analyzing the conclusion, Western blot was used to analyze the CAF and the CAF marker of alpha SMA that received the same treatment or 18 GY-shown. The results showed that the ability of the collagen gels to contract was not affected by the radiation regimen. As we can tell from the figures from the original article, it shows the influence of the radiation exposure to alpha SMA and other proteins like P53 and P21. So the first figure is for P53 and P21. Obviously, we can find that the results have a clear difference between the sham and different amounts of radiation. So compared with this, let's have a look the figure of alpha SMA. Among them, we can see that the sham as a negative control group didn't significantly differ from the results of receiving radiation. So we can conclude that the radiation exposure has no effect on alpha SMA expression. And the second finding is the CM to CFIR from irradiated CFs induce the IGF-1R and act phosphorylation in colorectal cancer cells. So Western blot is the most common method used for assessing the phosphorylation state of protein, and this research used this way to detect the phosphorylation of PIGF-1R tyrosine and the insulin receptor PINS beta after stimulation by CM-CFIR. As you can see, there are two bands on the figure, and when the upper band is darker, it symbols that the phosphorylation level is higher. The bands below serve as negative control group to confirm that the total protein mass of the samples is the same. It's obvious that under the action of CM to CFIR, it's displayed as a plus symbol, and the color is significantly deepened. We can also know from using the same research method, CM produced by injected CAFs increase tyrosine phosphorylation of IGF-1R and the insulin receptor in colorectal cancer cells. The experiment uses Western blot to compare the level of phosphorylation of IGF-1R and INSR, which is in CM to CFIR condition. The intensity of the bands corresponds to the phosphorylation level. So under the CM to CFIR conditions, increased band strength of the phosphorylative IGF-1R and INSR indicated increased phosphorylation. And then turn to the third key finding, the treatment of the CM, which was irradiated of the CAFS, resulted in changes of the expression of proteins involved in glutamine metabolism. So after 14 hours of treatment with CM to CF-CTR or CM-CFIR, use Western blot to test the proteins which associated with glutamine and metabolism and transport. The most supporting evidence in this test is that the amount of glutamine degradation products ANSN is increased. Since metabolic enzymes and FCAT transporters work together as a protective mechanism, when the amount of the metabolic enzymes changes, we can infer that their metabolism and transport protein are affected. So let's think about the picture in the same way we just say. With CM to CF-CTR as the negative control group, the expression of ASNS protein was significantly increased in the band stimulated by CM to CATIR. So as the final key findings, the IGF-1R and the INSR phosphorylation capacity of the CM, which is from irradiated CAFs, is equal to the specific concentrations of recombinant human IGF-1. So in order to detect the biological active and the potence of CAF's conditioned medium, it used Western blot to detect the phosphorylation level of IGF-1R and INSR, as well as the downstream active of AKT. So in the results, P to IGF-1R, P to INSR, and AKT, phosphorylations increased. The level of phosphorylation induced by CM on irradiated CAFS is compared to that induced by about 10 ng of male RHIF1, IGF-1.
Okay. Based on the contents provided in the previous, then it is safe to conclude that this research plays an important role in pinpointing the unintended effects of radiotherapy on the microenvironment of tumor. This is demonstrated in how radiotherapy could activate CAFs to enhance the proliferation of cancer cells and their survival. This research matters to society in that it enhances the understanding of complex links to the treatment of cancer for identification of new therapeutic targets. In other words, it highlights the need for the utilization of a myriad of therapeutic interventions that would go a long way in inhibiting detrimental effects as well as improve the prognosis and the rate of survivals for patients with colorectal cancer.
Tomline's experiments regarding radiotherapy's effect on CIFs and IGF-1R demonstrate that there are major shortcomings regarding the treatment of some cancers and that new therapeutic targets need to be found to effectively treat these cancers. In the case of colorectal cancer, the fact that preoperative radiotherapy assists the cancer's growth is clear indication that a new therapeutic target needs to be explored to help mitigate the cancer's effect on the patient without resorting to invasive surgery. Progression of the cancer being assisted by treatment also indicates that cancer treatments are not universal, and individual types of cancer likely require individual treatments. However, since this study does not touch on initiation of colorectal cancer, we are unsure about the internal or external factors that affected the initiation or initial progression of the cancer. The increased expression of IGF-1R and the downstream induction of anti-apoptotic signals is a hallmark of cancer, and an increase in this factor within an already existing cancer brings it that much closer to a metastatic cancer. For this reason, colorectal cancer and presumably other cancers that are high in cancer-associated fibroblasts such as breast or prostate cancer need to be targeted by researchers to discover new treatments that will prevent progress through growth factor overexpression.
For the limitation part, however, the Western blot used in this article still have some limitations. So the first one is that the result of Western blot is not as accurate as data. The data produced with a Western blot is semi-quantitative. So like what we talked about in the previous findings, we use intensity of bands to determine the samples. It can just tell us the sketchy results, not exact concentration. Secondly, the principle of the Western blot is to detect proteins through the binding and recognition of the antibodies to one or more targets. In this situation, the specific recognition sets should be highly specific. For example, in the detection of phosphorylated IGF-1R and INSR in this article, if the experiment didn't use non-highly specific antibodies, it can lead to misreading of the phosphorylation. So finally, Western blot is automatically detected in dynamic range, which makes the results misleading. In the study we researched, Western blot was frequently used to compare phosphorylation levels under different treatment conditions. If the amount of the phosphorylated proteins varies greatly, the dynamic range of the Western blot may not accurately reflect this difference.
So, based on the information provided by this article, and based on the findings of this study, next steps would include examining the possible inhibitors of IGF-1 pathway to compress the pro-tumorigenic impacts of radiotherapy. As such, additional research is required to validate the findings of this research as well as evaluate the therapeutic significance of utilizing multiple interventions to touch the IGF-1 pathway.